more effectively visualize this is looking at the superfamily. It began in 1985 with the first isolation of the glucocorticoid receptor, which provided a molecular template, which is shown on the bottom as a series of domains, and all nuclear receptors basically have this exact same domain structure. And at the time, since we were isolating a steroid receptor, we thought that we would be getting a small family of steroid receptors, ultimately 5 or 6 of steroid receptors. But in the end, what we thought we were going to get, there were 42 additional nuclear receptors that were unknown, and therefore 42 novel. But now we have a superfamily here, and it's up to us to both deconstruct it and then to use these discoveries for new types of therapies. So this is deconstructing colon cancer risk, revealing FXR as a therapeutic target, and that's really the principal goal of this talk. And why am I speaking of cancer risk in general, and why is this important? Let me briefly say that the discovery of the bile acids initially goes back to Adolf Windaus, and he won a Nobel Prize for his discovery of bile acids and their similarity structurally and chemically to vitamin D. So this was a complete surprise. Vitamin D is completely unexpected to be related to a sterol. And this opened up a whole new field because there are many types of bile acids in our bodies, at least 30 to 35 different species of bile acids. And what we learned, and that was the concept for decades and decades, and that bile acids were surfactants that allowed lipid to be absorbed into the body. And while that is correct, what wasn't appreciated is that bile acids are actually hormones. And that's what our discovery of the first bile acid receptor, FXR, in 1995 changed the field of bile acid physiology into one that is not just about transporting nutrients around, but rather is operating through a nuclear hormone receptor. That means changing genetic patterns throughout the body. Very different way to think about how this system works, and open up an entirely new approach to thinking about bile acids, what they do, and how they might be used to study disease mechanisms and therapy. And so what is actually inside the disease mechanism? So just to illustrate this in a simple way, if we look at these cogs that are interacting with each other, there's a hereditary cog, and that's if you have something like Lynch syndrome, something in your family, your risk is higher, and that begins to put pressure on growth. There's an environmental component, which, like the Western diet, the high-fat diet, that's also a key component here. And then the key thing for cancer is increased replication. And so somehow the risk with the familial link and the environmental link have to put pressure on replication to increase. And that is quite a bit of a challenge. But all these components are interacting with each other, and this is just a way to break down the nature of the system. And what are the links between H, E, and R? Hereditary, environment, and replication. And that's Pandora's box. We don't really know. And I want to use this as a transition, talk a little bit about the core theme here, the bile acid receptor FXR, which I mentioned earlier. And what you can see in this diagram here is that bile acids, there's over 30 of these species. They're synthesized in the liver, then delivered to the gallbladder, and when you eat a meal, they're released into the intestine. The nature of the bile acid receptor, the organs that are shown here: kidney, the adrenal gland at the top of the kidney, the liver, gallbladder, the intestine itself, the pancreas. These are all expressing very high levels of the bile acid receptor, FXR. So these are FXR target tissues. What does that mean? This is just a schematic of the FXR receptor interacting with a ligand. This is a classic bile acid that would bind to FXR, chenodeoxycholic acid, and then act now, as we believe, as a steroidal-like hormone, activating genetic programs in the intestine, liver, and other organs. We can then begin to explore this in some specific ways. What I want to specifically point out here is my lab created a form of a ligand for FXR that's a synthetic compound. This is a chemical that we made in the laboratory. It binds selectively to FXR, and in particular, this compound only acts in the colon. It does not get into the body, does not get into the bloodstream. And because of that, we're going to look at what is the impact of activating intestinal FXR with this drug called fexaramine, FXR amine. That was the title. So we call it FEX. And we'll kind of use this to explore a little bit about the nature of the signaling pathways. It's called the farnesoid X receptor, and that's because farnesyl is one of the direct synthetic precursors to cholesterol. This is in the cholesterol synthetic pathway from mevalonate going forward. And it's also very, very similar to juvenile hormone, the molting hormone in flies. And I should mention that the FXR receptor in humans is very closely related to the ecdysone receptor in insects. And juvenile hormone in insects, it controls molting. And so a lot of plants make juvenile hormone, and they do that to prevent insects from eating them. That juvenile hormone in the fly is acting on the fly equivalent of FXR, which is the ecdysone receptor. And FXR and ecdysone bind to the same exact sequences as well in very similar ways. So the point of this slide is to show you the complexity of the generation of bile acids, as is indicated here, both in what's called the neutral pathway and the acidic pathway. And this represents hundreds of millions of years of evolution to create this complicated biosynthetic theme. All these are steroidal-like hormones. So there's a lot of importance if they're going to have this kind of a pathway here. And we're very interested in the pathway, number one. Number two, what was not appreciated is that the synthetic rate at which you can process and produce bile acids has certain boundaries, and it can only produce so much in a particular unit of time. But this is the nature of the complexity that you have to deal about when you're talking about bile acids in general. So here are the questions we want to ask. For bile acids, they're linked to inflammation and colon cancer and IBD, as you'll see. What are the unsolved questions? So how does FXR impact intestinal homeostasis? This is key. How do bile acids modulate intestinal physiology? Pretty straightforward, but not actually when we get into it. How does high-fat diet promote intestinal inflammation? How does high-fat diet accelerate cancer progression? And is FXR a therapeutic modulator or a therapeutic target in colon cancer and IBD? And so, and we're going to keep looking at it in the form of hereditary, environmental, and replication components. And here we are using APC min mice, either on a normal diet or on a high-fat diet, as is shown here. And you can see on a normal diet, you have the adenoma, which is expected. But unexpectedly, this is the one that's not cancer. But in this case, you're going from adenoma, which is that first step that we saw with just the APC min mutation, to adenocarcinoma. So you have jumped over the RAS, MYC, P53, the whole, that slow progression of a change. We have leaped, and those changes occur over 20 or 30 years, we have leaped into immediately using diet, which jumps over all that and creates adenocarcinoma. And so this is unexpected. It's the first time that this has really been seen. And now you begin to understand the link between diet and cancer risk that we didn't fully appreciate. And you can actually visualize it here. Categorize these different components. We have genetics, like the APC min. We have the diet, like high-fat diet, that is sort of converging on changing to urocolic acid, deoxycholic acid, and other bile acids. We've profiled them all. Acting on FXR, and these altered bile acids almost universally are antagonistic to FXR activation, and that reduces FXR. Knockout of FXR is also very inflammatory, promotes leaky gut as well. And so there's a lot of different things in terms of the driver mutation and the diet, and the bile acid pool composition now and function that are coming into play as we use FXR as our guide to sort of unlock the pathway. And at the same time, I think that this opens a door to think about how we might be able to intervene by reactivating FXR activity. So I mentioned a little bit about some of the actions that are being produced in these various different states. This is one that I'll just briefly mention, looking at the production of FGF15 from in the intestine. This is one of the important regulatory factors. It's FGF19 in humans that is very highly controlled by the bile acid receptor FXR, and when it's activated, typically during a meal with normal bile acids, then FGF15 is produced and travels from the gut to the liver, and it controls bile acid synthetic processes. So this is a major regulator of bile acids, and what you can see here is in this that on a normal diet and on a high-fat diet, this critical regulator of intestinal function and the response in the liver is very dramatic. And just to point out, I'll just point out here, for example, FGF15 in red, which is the APC min component. That means it's you can't produce FGF15 basically. If you give the ligand, you restore, as is indicated here. And the same thing is true basically where you have red, it's being suppressed. Where you have green or yellow, you are seeing induction. This is really important. And again, this is rescuing in some very, very wonderful ways, depending on if we're using the adenoma or the adenocarcinoma diet. So the adenoma is the high-fat diet alone. This is with the mutation. And the effects are really quite dramatic. So what about getting back to aspects of cancer? And importantly, FXR, and looking at one of the key features here, which is leaky gut. And if we look at in the adenoma case, that is not on high-fat diet. So what we see is an APC, basically at no leaky gut with APC min. You have leaky gut, you can see the amount. With FXR activation, you really bring this down. If you look at intestinal permeability, an adenocarcinoma, same thing, just the numbers are larger. You have dramatic intestinal permeability. You're bringing it down with the drug to about 100. This gets you to 75 or so. So this is actually quite dramatic. And in general, this approach says that it might be possible to restore gut integrity in both the adenoma and the carcinoma model. So this begins to let us think about, might this really be a therapeutic? And so we explore this a little more in terms of how FXR impacts both the bile acid pool and function. And this is one of these just very dramatic results. I'll go over here in the adenoma case, which is wild type mice. There's very little bile acid in terms of what's in your bile acid pool composition, plus or minus the APC min mouse in diet. So there's not much to regulate. If you see what's happening in terms of in the adenoma component with APC is already driving up the BA pool. FEX brings it down significantly, at least by about 40%, not fully. But more surprisingly is in the adenocarcinoma, where we are putting the APC min mutation on high-fat diet, you're getting up to just a gigantic serum pool size. And in that context, you now see FEX-D bringing things down. Keep in mind that people who are obese and have an APC min mutation are going to be likely in a very dangerous state. This suggests that we could lower risk and also by giving us a mechanism, potentially a way of controlling the process. So FEX-D decreases serum bile acids, triggers this cascade of events that increases specific bile acid species. This led us to consider whether we could actually use fexaramine, intestinal-specific ligand, to treat these animals in both the APC min on normal diet and the APC min on high-fat diet. So these are all going to be APC min. And I should say that it's not an ideal model because in humans it's colon cancer. In rodents it's upper GI cancer, and they typically die from blockage. What I didn't mention is the APC min mice on high-fat diet do get colon cancer, but blockage is a challenge in this system. But either way, if we give FEX-D, we get an extension here from 30 to 50 weeks, and that shows conceptually that a gut-restricted activation of FXR is able to change a gene network. And we can measure that gene network, and we have. We've done the same in these two contexts. We've also worked with organoids, human organoids as well, to look at changes that we can establish gene expression changes by delivering the ligand or not. And so this then led us to, because we have the gene changes, we can take those genes and go into the database for humans with colon cancer and ask if you have the FXR gene expression that we have seen in human organoids, or the APC-min mouse, so human and mouse, if you have this gene set, you are likely to have a increased survival probability. And so that now is somewhat translating the concept of pathologic and physiologic convergence and taking away the pathologic state at least far enough to have an impact potentially on survival. And so this obviously opens up a way to begin to think about future studies and what we might be able to do. And to just quickly summarize, I'm calling it bile acids back to the future. It was one of the great stories 100 years ago. And the idea here is that bile acids are dynamic. Their chemical composition and functional properties vary across time, diet and genetics and disease. So they're very dynamic. The BA pools respond to external stimuli, food and the microbiome, bile acid species, number of nuclear receptors, not just FXR, but these other nuclear receptors are all good bile acid receptors. This one is an exception. This is a G protein coupled receptor, TGR5, also a good bile acid receptor. So we're learning so much more about bile acids than we ever knew, and that opens the door for this being a major new approach in physiology. And I call this a liquid organ. Because these are hormonal, having all the properties of a regulatory event, but the way in which they are passed from the liver, packaged in the gallbladder, and delivered collectively. I'm calling it a liquid organ as a way, again, to conceptualize the thinking about how this washes through our system and impacts on our body physiology. And so one way to begin to think about classifying these, this is very simple, is taking the known bile acids, let's show them here on the left, and simply stacking up to, are they activators of FXR? Are they repressors of FXR? Do they have or no have any activity at all? Do they impact the microbiome? Are they changing lipid production? Do they interact with vitamin D receptor, TGR5, any of these other receptors that I've already mentioned? How do they impact the APC-min pathway? And you can just increase the depth of this and begin to deconstruct something that has been around for 100 years, but now in an entirely new way. And keep in mind the networks that are controlled transcriptionally in target cells throughout the body are enormous. And that means that there's the way that you achieve this benefit is because of FXR and its distribution throughout the body. So deconstructing risk reveals an unrecognized mechanism of convergent pathways that deplete FXR signaling. This suggests that FXR activation by virtue of synthetic ligands, in our case just an intestinal ligand, could dually block the oncogenic and high-fat diet cascade to restore intestinal homeostasis. The current interest in my lab is homeostasis and enable a new approach to cancer therapy.